parasites are basically classified into two major groups. One is called as the protozoa. So these protozoa, they are being formed by single cellular organisms, and some parasites, they are formed by multicellular organisms. So among the different metazoan parasites, the helminths, they constitute the main parasites of the livestock, and the word helminths, they come from the Greek word helminthos. It means worms. So helminths are basically three groups: trematodes, cystodes, and nematodes. So today's our discussion is on the cystode parasites of goats. All cystodes, they belong to the phylum Platyhelminthes. So under this Platyhelminthes, there are two classes. The one is the Eucystoda, and they are called as the true cystodes, and second one is the Cotyloda. Cystodes, they are basically hermaphrodite. That means the cystodes or the tapeworms, they are male and female parasites are present in the same individual. That means the single parasite is having both male and female organs. And these cystode parasites, they are dorsoventrally flattened bodies, and they are elongated, just like that. And that is why these parasites are known as tapeworms. So a typical cystode parasites, they are consisting of three parts of the body. One is called as the head or scolex, and there is a short portion that is called as the neck, and the entire body, it is consisted of different segments, and these segments, they are altogether known as strobila. So coming to the scolex, so scolex or head of a tapeworm, it is a protrusible cone-like structures, and that is armed with the hooks. So in most commonly, many of the cystodes, they are consisting of two pair of hooks, and there is a circular structures, and these are known as suckers. So these are the different body parts of the head of a tapeworm. Then the neck portion, it is a small unsected portion, and the rest of the body, it is consisting of different segments, and these segments, they are also known as proglottids. So proglottids are three types. So those proglottids, they are anterior to the head or scolex. They are known as immature proglottids. That means these proglottids, they do not have functional reproductive organs. Then this is followed by the mature proglottids. So mature proglottids, they contain the functional reproductive organs. That means in mature proglottids will have both male and female reproductive organs, and the posterior portion of the proglottids, they are called as gravid proglottids, because these proglottids, they contain only the eggs, and whenever the gravid proglottids are full of eggs, they will be shed out to the outside, and from each of the proglottids, gravid proglottids, the eggs come out through a process that is known as apolysis. Then coming to the life cycle of cystodes. So we'll see what is the general life cycle of the cystodes. The egg of the cystodes, they are either embryonic or non-embryonic whenever they are coming out from the body of the host. The embryo inside the egg is known as oncosphere, and this oncosphere, it is a spherical structure, and it has three pair of hooks, and with these hooks, the parasite, when they start growing, so it is to firmly attached to the mucosal surface of the body of the host. The oncosphere, it is consisting of a membrane that is known as oncospiral membrane, and inside the oncospiral membrane we'll see the embryo that is known as the hexacanth embryo, and the surrounding this radiated structure, it is known as embryophore, and this embryophore, it protect the inside embryo from desiccation. So if this embryophore is thick, so it indicates that this ova or this cystoid egg is very resistant to desiccation, mostly to extremes of sunshine, because these eggs are being voided by the host in the environment, and in the environment, any harsh environment, it can withstand due to the presence of this thick embryophore layer. On ingestion by the intermediate host, the oncosphere get released by the action of host digestive juice, and then it penetrates the intestinal mucosa and reaches its site of predilection for development. So this outer layer is only dissolved whenever this egg is being ingested by the intermediate host, and then only this outer layer is dissolved and the inner hexagon embryo will be released for further development. Then in case of cystoid, the larval stage of the cystodes, it is known as metacystodes. And these metacystodes is usually developed in case of the intermediate host, because we can see that in case of cystodes, the life cycle is indirect. It always requires an intermediate host for completion of the life cycle. And this intermediate host varies from vertebrate host to invertebrate host. So depending upon the type of parasite, cystoid parasite involved, the type of intermediate host is also varied. Then we will see what are the metacystode or larval stages of cystodes that we can see in case of goat. So in case of goat, whatever the tapeworm infection we get, so some of the tapeworms, their larval stage is causing the disease, intermediate host, or in case of goat. Or some parasites, their adult stage is causing disease in the goat. So initially, we'll go for disease that is caused by the larval stage of different cystodes. So the first larval stage, that is known as the cystosarcoid. So it is a metacystode or the larval stage of the cystoid parasite, and this cystosarcoid, it is a pinpoint sized vesicle which once collects, not invaginated, without a cavity, and it usually develops inside the body of invertebrate host. So this type of cystosarcoid is found in case of Monagia infection in goat. So whenever we, in the subsequent slide, I will discuss on how this cystosarcoid is causing the disease. So let us go to the next slide. Then another metacystode is cystosarcas. So this cystosarcas, it is a bladder-like vesicle containing fluid, which once collects is invaginated and is usually develops in the striated muscles and other body parts of the mammals. So this type of metacystode is generally found in case of vertebrate hosts. So those vertebrate hosts, particularly in sheep and goat. So these metacystodes usually develops, and this type of metacystode, this cystosarcas, it is a single scolex vesicle and it contains the fluid, and these are generally seen within the body cavities of the intermediate host, particularly in case of sheep and goat. So in the omentum or in the mesentery, or they may be attached to the fascia of the any internal organs. So these can be visible, and these metacystodes are seen only at the slaughter or necropsy, when such intermediate host or the sheep or goat, when they are being slaughtered, so these metacystodes can only be visible, but from outside it is not visible. There will be no any clinical symptoms. So whenever the carcass is open, then only these metacystodes are visible. The next metacystode is called as the cynoras. So this cynoras, it is a thin semi-transparent bladder and contain plenty of fluids with a large number of colices invaginated from the germinal layers. So this is the single germinal layer, and inside which we can throw with naked eye, one can see the scolices. So these are nothing but the protoscolices of each of the tapeworm parasites, and these are generally arranged in clusters, just like our grapes. So these are being arranged in clusters, and very easily one can see these protoscolices with naked eye, and these are on magnification. Either through zooming through our mobile phone, also one can see the these protoscolices. It's just like a button type of structures or small gram. So these are the protoscolices one can see with the naked eye. So these metacystodes also develop inside the body of intermediate host. So that intermediate hosts are the vertebrate hosts. Then another metacystode that is known as the hydatid. So it is a thick walled, large sized bladder filled with fluid, producing brood capsules from the germinal layer, and inside each brood capsule, the number of scolices develops. So there is a difference between Cynorus and hydatid. So in case of Cynorus, it is also a fluid-filled cavity, but its terminal membrane is very thin. But here, in case of hydatid, the layer it is thick, the outer layer is very thick, and inside which we can see the scolices, and they are being surrounded by the thick capsule, and these are known as brood capsule. And inside each brood capsule, we can see number of scolices. They are vegetated from the germinal layer, and these are the pictures showing the hydatid cyst, each of the hydatid cysts, and on opening of these cysts, we can see number of scolices they are found inside each of the vesicles. Then coming to the epidemiology of metacystode infection in intermediate host. So this metacystode infection or larval infections in intermediate host. So how this intermediate host they get the infection? Because we can see that in case of goat, we see two type of infection. So some cystode parasites, their larval stage is causing the disease, and in some cystodes, their adult stage is causing the disease. So we'll first of all we'll see that what are the larval stage of parasites, how they are infecting the mammalian host, that is the goat, how they are infecting in the host. The prevalence of infection in intermediate host is affected by the level of environmental contamination, dispersion of eggs arrival, age of the host, and immune response of the host. So let us first see the level of environmental contamination. So how environment get contaminated. So this may come from the infected definitive host. Infected definitive host, they passes the proglottids through feces that contain the infective eggs, thereby act as a source of infection. They contaminate the feed, water, as well as the pasture land. And many of the cases in metacystode infections, we can see that the definitive hosts are the canines, and these canines or the dogs, they used to set the gravid segments in their feces, and these gravid segments containing the eggs. So when they are being voided in the environment or particularly in the grazing land, so they will contaminate the grazing land, and whenever the intermediate host, particularly the sheep and goat, when they come for grazing, they used to get the infection. So during grazing, they also ingest the eggs, and thereby they become infected. And pasture contamination by infected stray dogs onto the pasture land. So especially the stray dogs. So those who are domestic dogs, they are regularly being dewormed. But those which are stray dogs or roadside dogs, they used to continuously get infection from the environment, and they are shedding the eggs in their feces, and they contaminate the entire grazing land. So pasture contamination, the type of if the pasture is more heavily infected with the eggs of the parasite, so chances of getting infection by intermediate host is also increases. That means it is directly related. Infection of the intermediate host is directly related to the contamination or contamination of the grazing land by the infective definitive host. Then second factor, presence of suitable susceptible host. So susceptible intermediate hosts are exposed to infective eggs. They pick up infection and thereby harbor the metacystoid stage of the parasites, and these are the infective stage for the definitive host. So once the intermediate host, they pick up infection from the environment, the parasite, the oncosphere, so it gets released and it will pass through the blood vascular system. It will reach to its site of predilection, and the metacystoid will be developed in the in the site of predilection, and thereby the infective stage of the parasites will be developed inside the body of the intermediate host. And if such intermediate hosts are being slaughtered, or somehow if that cyst is being ingested by the definitive host, so infection will be perpetuated in the environment. Then third factor is the dispersion of egg on pasture. So most commonly the tinea eggs, they can travel up to 80 meters under suitable environmental conditions. Because in case of tinea eggs, so when the segments are being voided, when the crevice segments are being voided by the definitive host, the segments, they have some movement, they have some crawling movement, and these segments are being disintegrated, and these eggs, they can also have little bit of movement, and with that the eggs will be available, or eggs will be totally dispersed in the environment. And also this movement is also favored by the wind. And secondly, these eggs are also being dispersed through some insects like flies, beetles, earthworms. So flies also transmit the eggs to distant places, and floating water, especially in flood water. If the grazing land is flooded, so the flood water is also helping dispersion of eggs to a distant place, and thereby infection is also being perpetuated. Then fourth factor is the egg survival. So normal condition survive up to 6 months, but some eggs, so if the eggs are being laid long back, and if these eggs are not being ingested by the intermediate host, so these eggs, they will lose its viability, and thereby also the infection can be reduced. But thing is this, so if these recently voided eggs are being ingested by the intermediate host or the goat, they may capable of becoming infections. Then age of intermediate host. So infection to the intermediate host, particularly the goat kids, so up to below 5 weeks of old, so they are found to be less commonly infected, because the maternal antibody will prevail, and that will give protection to infection, or it will give resistance, some kind of resistance to infection. But those kids which are more than 5 weeks old, so maternal immunity starts declining, and at that time, if these animals are being allowed for grazing in a heavily contaminated pasture land, they may become infected. And kids, those reared on contaminated pasture land from birth onwards, so they will develop strong immunity against to reinfection. So these are the factors that help in particularly the age factors of involvement of infection in intermediate host or the goat. Then immune response of the host. Animal develop a marked protective immune response to reinfection with metacystosis. So once animals are being infected with the eggs of the cystodes, so some kind of immunity develops in the host, and they can subsequently resist for infections. Then heterologous infection, so cross protection is observed between the tinea species. So sometimes what happens, so one tinea species, tinea eggs infection can give protection to against tinea infection. Mostly we can see in tinea multiceps infection against tinea haeditigena infection. Especially if a goat is being infested or infected with tinea multiceps eggs, So it is found to be somewhat resistant to Tinea hydatidina infection. Then let us go to the bladderworm infection in goat. So here in this slide, we'll see what are the bladderworms that commonly infect the sheep and goat. So although these parasites, they do not occur in goat, but they are metacystod or the larval stage of the parasite. They are causing disease in the host. So the first parasite is the Tinea multiceps. So the definitive host of this parasite is the dog, wild carnivores, as well as the human. So humans rarely may get infected, or accidental infection may occur in case of human being. And the site of predilection of the adult parasite is the small intestine. So here in this case, the intermediate hosts are the sheep and goat, and the metacystod or the bladderworm, that is known as Cynara cerebralis, and this metacystod usually develops in the brain and spinal cord of the goat. Then next parasite is the Tinea multiceps. So this is the parasite. So sometimes these Tinea multiceps parasites, instead of occurring in the brain and spinal cord, it is found to occur in the subcutaneous tissues or as well as in the muscle tissues. So that is why this type of cyanosis is known as non-cerebral cyanosis or subcutaneous cyanosis or muscular cyanosis. So the definitive hosts are the same. The site of predilection of the adult parasite is also same. Intermediate host is the sheep and goat, but the metacystodes that cause which occurs in the subcutaneous tissues and muscles, it is known as Cynoras gregeri. Then next parasite is the Tinea hydatidina. So it occurs in dog, wolves, and other wild carnivores. The parasite usually develops in the small intestine. Intermediate hosts are the sheep and goat, and the metacystod that is produced from Tinea hydatidina, it is the Sathyazerograzing tenuicalis, and these metacystodes usually develops in the vital organs, and very commonly it occurs in the liver and other abdominal cavity, particularly in the omentum or in the abdominal cavity near mesentery. So there these metacystodes usually develops. Then Tinea ovis, this parasite very rarely occurs in dog and other wild carnivores. Then there is very scanty reports on prevalence of Tinea ovis in dog and other wild carnivores. The site of predilection is same with the other parasites. It is the small intestine of dog and cat, where the metacystodes they develop into the adult stage of the parasites. The intermediate host are the sheep and goat, and the metacystod, it is known as Sathyazerograzing ovis, and this metacystod usually develops in the skeletal and cardiac muscles of heart as well as in the diaphragm. Then next bladderworm, it is the Echinococcus granulosus. So that cause the disease condition that is known as cystic echinococcosis, and the definitive hosts are the dog and other canines. The site of predilection of the adult parasite, it is the small intestine. The intermediate hosts are the herbivores, and mostly the sheep and goat, they may harbor the parasites, and this parasite is very commonly occurs in case of human being. This disease has got zoonotic importance. Here man also act as an intermediate host, where there is development of hydatid cyst in the vital organs, and among different vital organs, liver and lung are the most common site for development of the hydatid cyst. So one by one we will discuss the different metacystod infections. Now we'll little bit discuss on the cyanosis or GID. So cyanosis, or also known as GID, tardy or staggers. So these are the different synonyms. So it is a metacystodal infection of ungulates, especially sheep and goat, caused by invasion of the brain and spinal cord by the intermediate stages of the parasite Tinea multiceps, and these intermediate stages, they cause the formation of the metacystod that is known as Cynara cerebralis, and these metacystod, they invade the CNS, and the different syndromes produced. It is one of the localized space occupying lesions of the central nervous system, and that cause focal encephalopathy. So since the metacystoid develop in the brain and spinal cord, so neurological symptoms we can see, as well as there is affection of the brain, and that condition is known as encephalopathy. The parasite is the Tinea multiceps. Definitive hosts are the dog and other wild carnivores. The site of predilection of the adult parasite, it is the small intestine of dog, particularly in the level of jejunum and ileum. We can find out the presence of adult parasite attaching to the mucosa of the jejunum and ileum. And the intermediate host of this parasite is the sheep and goat. And this metacystoid, it is known as Cynorus cerebralis. And the site of predilection of the metacystoid, it is the brain and spinal cord. So here we can see the life cycle of Tinea multiceps. So dog acts as a definitive host. The adult tapeworms develops in the small intestine of the dog, and when these adult parasites develop, so it will start forming eggs, and these eggs are available in the posterior portion of the proglottis, which are known as gravid proglottis. And these gravid proglottis, they are devoid of any reproductive organs. They are fully consist of the eggs, and these gravid segments, time to time, they are being voided by the definitive host, and these are being voided if these gravid segments are voided in the open grazing system or in the grazing land. So during grazing, the intermediate host, as we know, the intermediate hosts are the sheep and goat, so they will these eggs will be ingested by the intermediate host, and following entry of the eggs into the intermediate host, the outer oncospheral layer, it is being dissolved by the host digestive juice, and this oncosphere, they will be released and they will penetrate the small intestinal mucosa, and via circulation, so they will be migrating towards its site of predilection. And in case of Tinea multiceps, the site of predilection of the metacystoid, it is the brain and spinal cord. So the metacystoid usually develops in the brain and spinal cord, and we can see the development of metacystoid in the brain, and this metacystoid, it is known as Cynorus cerebralis, most commonly in the sheep and goat. But among sheep and goat, sheep is most commonly infected as compared to the goat, where we can see the development of the metacystoid. So this is also a graphical representation of the life cycle of Tinea multiceps. So the parasite, from this metacystoid, the adult parasite will be formed. Then this adult parasite, so it will shed the eggs, it will form the eggs, and these eggs are being ingested by the intermediate host. So intermediate hosts are nothing but the sheep and goat, and this following ingestion, the metacystoid will be formed either in the brain, or sometimes these parasites, they have a tendency to develop metacystoid in the subcutaneous as well as in the muscular tissue. There also we'll see the formation of the metacystoids, and rarely, sometimes, if the human, they are being ingested, if they accidentally ingest the eggs of Tinea multiceps, so the metacystoid also develops in the brain, or particularly sometimes they may also affect the eyes, causing ocular cyanosis. So this condition also occurs in case of human being, and these animals, when they are slaughtered, we can see the development of metacystoid in the brain. And the dogs, particularly the stray dogs as well as the wild animals, because we see in under Indian condition, many of the slaughterhouses, they are not being constructed on scientific manner. So in normal market area, we can see there is dogs, they will surround the slaughterhouse, and if the butchers, they found the presence of any metacystoid, they quickly they throw the metacystoid, and these roaming dogs, these free roaming dogs, they will quickly eat the metacystoid and they may get the infection. So once the definitive host, they eat the metacystoid, so the adult parasite will be developed in the small intestine, and that way the life cycle continues in both definitive host as well as in the intermediate host. And accidentally, the human may get the infection, and when women become infected, so metacystoid will be developed in the brain as well as in the eyes, and they may cause neurological syndromes. So in short, life cycle already we have discussed. So intermediate host is infected through ingestion of Taenia multiceps eggs. So each egg will contain an oncosphere that hatches and is activated in the small intestine. The oncosphere penetrate the mucosa. They are being carried via the bloodstream to the brain or spinal cord. In goat, the cyst can form in subcutaneous and muscular sites as well as in the brain and spinal cord. The oncosphere develops into metacystoid larval stage that is known as Cinera cerebelis, which measures into thin-walled fluid-filled cyst in about 5 cm in diameter. The Cinera cerebelis measures into thin-walled fluid-filled cyst. The life cycle is completed when the canine eat the raw infected brain or spinal cord or offal containing the Cinera cyst. The scolex or the head of the tapeworm, they embed itself into the wall of the small intestine and begin to grow, become adult, and start shedding the eggs. So this is the graphical representation. We can see the development of Taenia multiceps oncosphere into the mature oocyst and related phases of intermediate host infection. So following infection into the intermediate host, so within 8 to 10 days, the eggs, they are being released. The oncosphere is the outer cover oncosphere is released, and they will start migrating to the central nervous system in 10 to 33 days, and in the CNS, some pyriform vesicles will be formed of 5 to 8 millimeter in diameter. So by 33 to 40 days, these pyriform vesicles will be formed, and by the end of 2 months, a seri-like vesicle is formed. It is about 0.7 to 2 cm in diameter, and by 3 months, we can see the development of the protoscolixis in the Cinera cyst, and by 7 to 8 months from infection, from the day of infection up to the development of a fully mature cyst, it takes around 7 to 8 months of time. And at this stage, the diameter of the metacystoid will be 5 to 6 cm, and on average, there will be 22 to 23 cluster of protoscolixes. That means one cluster will contain around 20 to 23 numbers of protoscolixes, and these clusters are nothing but the protoscolixes of the parasite, and thereby this metacystoid will be ready for infection to the definitive host. So at this stage, if this animal is slaughtered, so the metacystoid, if it is being ingested by the definitive host or the dog, they will become infected. So inside their body, the adult parasite will be developed. So there will be 4 phases of the infection inside the intermediate host. The first one is the invasive stage, 8 to 10 days. Then there will be acute cyanosis, 10 to 33 days. Then there will be a quiescent stage. So at this quiescent stage, there will be development of the protoscolixis. And in the chronic phase, so a full-grown metacystoid will be formed inside the brain or in the spinal cord of the intermediate host. Then coming to the pathogenesis. So following infection, so as there is infection in the brain as well as spinal cord, so the affected host will show neurological disturbance. And what are the pathogenic syndromes? So we can see the one is the meningoencephalitis. Here the metacystoid traumatize the cells of the brain, producing inflammation and cause the condition meningoencephalitis. There will be inflammation of brain and meninges. And anemia and atrophy of the organ. If the cyst obstruct the blood vessels, it results in anemia and atrophy of the organs. Especially if the cyst is grown on a blood vessels of the brain, so the supply to that particular structure of the brain So that will be hampered, and that will lead to atrophy of the organ, as well as there will be no blood supply to the organ because of compression of the blood vessel, and that will also cause to anemic condition. Then convulsions. The presence of cyst, particularly in the locomotor as well as in the non-locomotor areas of the brain, so it causes disruption of the action, and that will result in convulsions. Then fourth will be atrophy of the cranial bones. It occurs when cyst is situated on the upper part of the skull. So if it is grown on the upper part of the skull, so growth of the cranial bone will be hampered, and that will lead to atrophy of the cranial bones. Next, we see the clinical symptoms as per involvement of different parts of the brain. So in case of cerebellum, when there is growth of metacyst in the cerebellum, we can see hyperesthesia as well as jerky or staggering gait of the animal. If the cyst grow on the cerebral hemisphere, we can see tilting and circling towards the affected hemisphere. That is, tilting means the continuous shaking of the head, as well as circling movement we can see in the affected portion of the cerebral hemisphere. Then spinal cord. If the cyst grow on the spinal cord, we can see progressive paraplegia or quadriplegia. We can see. Then anterior brain. If the cyst grow on the anterior part of the brain, the head held against the chest, and there will be high stepping gait. So these symptoms. So if one can see these type of symptoms, we can clearly suspect that the cyst is being growing on this particular organ. I think the field bats will be benefited. So by seeing the symptoms, or one can suspect that the cyst might be growing in these structures. Then coming to clinical findings. So the clinical findings of cerebral cyanosis, it can be classified into three categories or three stages. In the first stage, the animals are depressed and show intermittent head tilt. There may be short, intermittent convulsions or circling, and these phase usually last for 4 to 8 days and may be overlooked. And many of the farmers, they may overlook these symptoms, or maybe it is being overlooked by veterinarians also, or generally it goes unnoticed. The second stage, it begins from 8 to 16 days after onset of signs. The most common findings are head tilt, unilateral blindness, circling, and loss of balance with staggering gait, forward walking with head held high, followed by head pressing against obstacles. So this, in second stage, we can see the typical clinical symptoms of cyanosis. In the third stage, so this begins from 12 to 25 days after onset of signs. Animals become recumbent, exhibit extensor rigidity with paddling movement of hind limbs. So by the third stage, the locomotory portion of the brain is affected, and that is why we are getting paddling movement of the hind limbs. And the animal is no more can get up, and it become recumbent and may die at this stage. So if the cyst is not removed immediately, the animal may collapse and it may die also. Then coming to lesions of cyanosis, so cysts mostly found in the parietal regions on the surface of the cerebral hemisphere, just behind the two horns. So clinically, so if we can suspect, so this triangular area just behind the two horns, so we can draw some imaginary lines. So this is the place, this triangular area is the area where we can feel for presence of the cyst, and if we feel some softening in this area, this triangular area, we may suspect that the cyst may be growing superficially. And this is the photograph showing. So based on the suspicion, if we open the skull, if we open the skin and open the skull, we can get the developing metacystoid from the brain. So this is the photographs. Actually, this are the photographs from my research work. So this, while removing the metacystode from the brain, so we have done a V-shaped incision. So V-shaped incision is being given, and just after putting light incision, the cyst, so because of the pressure, the cyst, it may hidden, and whenever it gets a small opening, so quickly the cyst is to come out, and with naked eye we can see the presence of protoscolicer, and that are arranged in clusters. And here also, after removal of the entire brain, so we can see the attachment of the cyst in the brain tissues from a goat. So this was also a little bit from my research work. So in this work, I have given experimental infection of metacystodes to a dog, and after a pre-patent period of about 60 days, there was development of adult parasites in the small intestine, particularly at the level of jejunum and ileum. We could find the development of the adult parasites, and these parasites, they occlude the small intestine, and these are the attachment sites where the parasites they firmly attach to the mucosa. For information, I can tell that these parasites, these cystode parasites, they do not have functional digestive system. They always depend on the half digested food. So half digested food are available only in the small intestine. So that is why many of the tapeworms, they sit, especially in definitive host. The site of predilection of the adult parasite is always small intestine, because in the small intestine they will get half digested food, and with that half digested food they draw nutrition and they survive. So diagnosis of cerebral cyanosis is done based on the clinical signs, those signs that have been already discussed. Then confirmatory diagnosis is done by firm digital palpation of the parietal bones just behind the two horns. And when we give pressure on this area, parietal bone area, so the animal results in bleeding. So maybe they feel pain, and that is why they bleed. And we also feel the softened areas of the skull, and thereby we can suspect that there might be presence of superficial cyst. But we have to see, so if the cyst is superficial, then only we can palpate the softening. Otherwise, if the cyst remain hidden inside the skull, or sometimes the cyst, if the cyst is not fully matured, so the softening may not be felt. And in that case, even if the animal showing symptoms of cyanosis, we can go for radiography, so that will detect the presence of the cyst. And also we can go for cerebral angiography, so these two we can give it. And radiography is always helpful if there is facility of radiography. So in especially for deep seated cyst, we can advise for radiography. Then coming to treatment for cerebral cyanosis, there is no effective treatment. So only treatment is surgical removal of the developing cyst. So if the animal is showing symptoms, but if the cyst is not removed surgically, the animal may die. So it is always advisable. We should advise the farmers to go for surgical removal of the cyst. And many of the times, sometimes the owner may not cooperate. He may not be interested to go for surgery. So in that case, we can give albendazole therapy. Albendazole recently was found to be highly effective at 10 mg per kg body weight orally daily for 3 days. So albendazole, it will kill the developing photoscolysis, as well as it cause calcification of the cyst, and thereby there will be regression of the cyst, there will be calcification of the cyst, and at certain times the host immunity, it will engulf the cyst and animal can be saved. But if the cyst is fully grown, it is not possible to eliminate it. So better we should, if the animal is showing severe form of the disease, or if the animal is in recumbent position, so better we should suggest for surgical therapy. But if the cyst is not palpable superficially, we can advise for albendazole therapy. Besides albendazole, we can also advise for combinations containing fenbendazole and praziquantel. Mostly we can use some commercial preparations like Fantas Plus tablet. So it is found to be highly effective. So fenbendazole and praziquantel combinations are always effective for eliminating tapeworm infection, mostly metacystode infection in goat. Then we'll see the subcutaneous or muscular as well as non-cerebral cyanosis. So the parasite, it is same, the Tinea multiceps, but here in this case, we can see development of this metacystode in the subcutaneous tissues as well as in the muscle tissues, and that metacystode, it is known as Cynorus gageri. Initially, there was confusion that whether this parasite is same, whether this parasite is Tinea multiceps or some other parasites, they are occurring in the subcutaneous tissues. But recent studies, it was found that based on the molecular findings, it was found that the parasite is same, it is the Tinea multiceps, but sometimes this parasite, they change their site of predilection. The metacystode, they change its site of predilection. So if the metacystode, they get the opportunity, so they is to develop in the subcutaneous tissues, and thereby this metacystode is developed, and that is known as muscular cyanosis or subcutaneous cyanosis. But if it is grown in the brain, it is known as cerebral cyanosis. But ultimately, for both these metacystode, the parasite is the same. It is the Tinea multiceps, and metacystode is known as Cynorus gageri. And there is another difference is that in case of definitive host, so although the adult parasite develop in the small intestine, but our experimental work on the dog, it shows that when dogs are being fed with the metacystode from subcutaneous cyst, so the adult parasites develops invariably only in the jejunum, not in the ileum. The adult parasites was invariably found in the jejunum instead of found in the jejunum as well as in the ileum. So this is the life cycle pattern. Only thing is that the metacystode, it usually develops in the subcutaneous tissues of the goat, and the other mode of infection to the definitive host, it is the same. The dogs, they get the infection through ingestion of the offals or the ingestion of the metacysts from the intermediate host. They become infected, the definitive host or the dog, they get infected, and the adult parasites develop in the small intestine. So these adult parasites, they will shed the eggs, or they will shed the gravid segments or proglottids that containing the eggs, and these contaminate the grazing land. And in the grazing land, if that grazing land and the intermediate host or the goat are there, so they will become infected. And following infection, the oncosphere, they penetrate the intestinal mucosa, they will be passed through the blood vessel, and they will reach its site of predilection, that is skin and subcutaneous tissues, where the metacystode invariably grow. And if these metacysts are being ingested by the definitive host, the definitive host, they will become infected. So this is the life cycle of Tinea multiceps gageri. So the name gageri was being given by the scientist Geiger, who first recognized the presence of the metacystode in the subcutaneous tissues, and that is why, in honor of Geiger, so the metacystode, it is known as Tinea and the Cynorus gageri, the parasite of which the adult parasite is the Tinea multiceps. And next we'll see how we can detect the presence of subcutaneous or non-cerebral cyanosis. So in case of subcutaneous or non-cerebral cyanosis, we always get a history of presence of grossly visible swelling anywhere on the body surface, which gradually increases in size. So owner always complained that there is development of some swelling on the body, and it go on increasing in size. And based on the history, we can go for clinical examination, and the clinical examination will reveal palpation of painless, fluctuating swelling, and there will be distinct dimension and the aspiration of the fluid. So it indicates a clear straw colored fluid, and that will be, and based on that we can diagnose it to be a cyst. And the clinical findings of subcutaneous cirrhosis is that in the initial stage there will be no significant symptom we can see, but if the animal is heavily infested with the subcutaneous metacystosis or subcutaneous cirrhosis, so we can see the clinical symptoms like lameness. So if the cyst grows on the thigh area or in the leg area, so we can see the lameness, then paralysis, and development of large skin lumps due to the subcutaneous. So we can see the severe lameness, and these cysts, they may sometimes cause pain and functional weakness in the involved organ. Because of the growth of the larger cyst, the animal feel difficulty in walking, so it shows lameness. The non-cerebral cirrhosis is observed mostly in the slaughterhouse. So although it is being so, but generally this type of cyst we can see, but owner never advised for any removal. But if that animal is slaughtered for meat purpose, there only we can see the presence of this non-cerebral cyst. Then how to recover the non-cerebral cyst? The affected animal is controlled manually on lateral recumbency. Then we can infiltrate with local anesthetic. So a vertical skin incision is given, and very easily we can recover the cyst, because the cyst is situated just below the skin in the subcutaneous area. So not much surgical intervention is required. And once the cyst is being exposed, it is cut, and the antibiotic powder is sprinkled on the wound, and skin sutures are being done, and that way we can remove the cyst. And this is the photographs of a non-cerebral cyst of from Skininfectionsinsheep. So we can, so with the naked eye, one can see the appearance of the protoscolices that are being developing, and it is arranged in clusters, and it contains all straw colored fluid, and this fluid is extremely hypertonic. So if we put the cyst in normal saline, so after some time the fluid will be withdrawn from inside the vesicle, and we can see there is shrinkage. So after putting the cyst in the normal saline, so as it contains hypertonic fluid, so the fluid is withdrawn and there is shrinkage of the membrane. And this is in the hyper magnification, especially under the stereoscopic microscope, we can see the protoscolices just like a button-like structures. And if a single protoscolis is being examined under the microscope, we can see this type of structure. So this is the scolex of the developing protoscolex. We can see these are the 4 suckers, and we can see the developing hooks, 2 pairs of in the rostrum, we can see 2 pair of hooks, and with the presence of these hooks, the adult parasites, they firmly attach to the mucosa of the small intestine of definitive host, and they used to draw nutrient from it. In one of my experiments, so I have experimentally infected some healthy dogs with the metacystosis that I have collected from non-cerebral cyst, and after a pre-patent period of about 60 days, we can see the shedding of the gravid segments in the feces, and when we open the intestine, we could see the intestine is almost packed with the adult parasites, and these parasites are invariably present in the jejunum of the definitive host or the dog. Then coming to treatment, so there is no effective treatment. Surgical removal of the cyst is the only way. Then albendazole therapy, we can use it for small ruminants, and in case of dog, we can also go for deworming of the dogs with combinations like fenbendazole and praziquantel. So thereby we can reduce the infection load in the dog. Then coming to prevention and control, so reducing stray dog population and restricting them from feeding small ruminant carcasses and offals. And very commonly we can see that in the slaughterhouses, those slaughterhouses which are not constructed scientifically, so we can see the butchers when they start cutting the meat, so the stray dogs they will surround, and they, if they get the access to the injection of these metacystos, the dogs they will become infected, they will perpetuate the infection in the grazing land, and thereby intermediate host will become infected. So that is why, so reducing stray dog population and restricting them from feeding small ruminant carcasses and offals, we should restrict them from entering the slaughterhouse. So that is why we should suggest for constructing scientific slaughterhouse, so that these dogs they may be less commonly infected. So as the life cycle of the cystoid parasite is complex, so our aim of control is by breaking in the life cycle of the parasite. So if we can break the life cycle of the parasite, then only we can control the parasite propagation. Then correct disposal of contaminated carcasses to prevent access of dogs to them. So if the carcass is found contaminated with the metacystode or the Cinera cyst, then we should properly dispose the Cinera cyst so that it will not be consumed by the dog. Then creating awareness among farmers about proper disposal of contaminated carcasses, as it is a disease having public health importance. We can see in case of Cinerasis, particularly in case of Cinera cerebralis infection, we can see the human, they may become infected. So we should aware the farmers through some awareness programs, through education of people. We should give trust on the importance of the disease in relation to the infection to livestock as well as to the human being. So we should always advise them to go for deworming, regular deworming of the dogs, then preventing the livestock in grazing in contaminated grazing land. Then we should prevent the stray dogs from entering into the slaughterhouses and prevent them from feeding of the carcasses or the offals having the metacystodes or the Cinera cyst. So those things we through some training or through some awareness program, we should tell to the farmers so that they can take the precautions, and thereby we can control this type of infections. Then regular deworming of dogs with praziquantel. So praziquantel is considered as a drug of choice for tapeworm infection in dogs. So intestinal and cystoid parasites can be controlled through strategic deworming of the dogs with praziquantel, particularly just before the onset of monsoon season, as well as in the post-monsoon season or in the autumn season. We can go for deworming of the dogs. So if we control infection in the dogs, no more infection, no more infected eggs will be available in the grazing land, and thereby we can also protect the animals, protect our livestock from becoming infected. So that way, by breaking the life cycle, we can only control the infection. In case of human, the metacystode of Tinea multiceps, they usually develops in the brain, or sometimes cyst, they may also infect the eyes, causing the disease condition that is known as ocular cyanosis. And when there is development of cyanosis in the eyes, so they will cause impairment in the vision. So many of the cases in our Sankardev Netralaya, we are getting the cases of ocular cyanosis. So there was some papers citing the publication of ocular cyanosis cases in human, and these are being found because of the playing of the pups. So if the animal, so our human beings, they have some affinity towards keeping a pet, but if that pet is not regularly dewormed, so infection may come, and thereby human may also, there is also chance of human infection. So people should be aware, or we should aware our pet owners about the importance of the disease, importance of regular deworming in canines. So those things we should tell to the owner So that then only we can control this type of metacystoid infection also in case of human being. The next disease is the cysticercosis or bladderworm of sheep and goat. So cysticercosis or bladderworm disease of sheep and goat, it is also a metacystoid infection. Mostly we see this disease in case of sheep and rarely in goat, and it is caused by invasion of liver and other vital organs by the intermediate stage that is known as Cysticercus tenuculus of the Tinea parasite, that is known as Tinea hydatidescena, and this adult stage of this parasite invariably occurs in the human, dogs and wolves, as well as in case of wild carnivores. So mostly this type of infection we can see whenever there is the fringe areas between the wildlife area and the fringe areas. That means the grazing land where our domesticated animals goes for grazing. So in the fringe areas, the wolves as well as the wild carnivores, they may come for the hunting of the small ruminants, and while doing so, these animals, these carnivores, the way they act as a definitive host, they will shedding or they void the feces, and if these animals are already infected, so they may shed the infective eggs, and thereby through ingestion of these infective eggs, so our livestock, our sheep and goat, they may become infected. So these type of infections we mostly seen in the fringe areas where the grazing land is shared by both wildlife as well as in the domestic animals. This cysticercosis, it caused significant economic loss due to condemnation of infected meat and offal, because the metacystosis usually develops in the vital organs, in the liver as well as in the other organs, and if there is development of the metacystosis in these organs, so there will be condemnation of meat and the farmer will face huge economic loss. So this is the economic importance, and this is the life cycle of Tinea hydatidescena. We know the dog and other wild carnivores, they act as a definitive host, and they is to void the gravid segments of the parasite Tinea hydatidescena, and these eggs are being ingested by the intermediate host, particularly sheep and goat, and following ingestion, so the oncosphere is released, and these oncosphere, they migrate towards its site of predilection, and the metacystosis is formed in the vital organs, mostly in the liver, and this metacystosis is known as Cysticercus tenuculus. And these metacystosis, if the animal is slaughtered, and we may find the development of the metacystosis in the liver, and by ingestion of these metacystosis, the definitive host, they will pick up the infection. And human, there is also possibility of human infection. So human infection may get while playing with the pets. So if the human being, they feed their food with the contaminated hand, because these animals, they should continuously shed the gravid segment if the nails are not clean. So, and if he is not maintaining the hygienicity before taking food, so the infective eggs may be ingested by the human being, and the metacystosis will also be developed inside the liver of the human being, and thereby the disease develops. So these are the different metacystosis of Cysticercus tenuculus that are being recovered from the carcass of the goat. So these are the metacystosis in the peritoneal cavity, particularly in the omentum we can see, in the fascia we can also see the development of the metacystosis, and these are the recovered cysts. We can see the Cysticercus tenuculus. So Cysticercus is nothing but the single invaginated scolex, and that is being invaginated deeply into the field cavity. But difference with the Cynorus is that in case of Cynorus, the cavity contains many scoleces, but in case of Cysticercus, the single scolex is invaginated. So that is the difference between the metacystosis of Cysticercus as well as Cynorus. Then mode of transmission in case of Cysticercus tenuculus, how infection transmitted. The intermediate host or the sheep and goat, they pick up the infection during grazing in an contaminated land defecated by an infected definitive host. These are canids as well as wild canids. And the definitive host, they pick up infection due to ingestion of the meat or offal infested with the metacystode of Cystosarcus tenucolis. So these are the metacystodes that are being recovered from the carcass of goat. This is the Cystosarcus tenucolis metacystode recovered from the liver of the goat. Then this is from the lung tissues. The metacystode is developing, and this is around the, so this is the trachea. So around the trachea, the metacystode of Cystosarcus tenucolis is being recovered. Then diagnosis based on detection of gravid segments in the feces. So this is the strobila of Tinea hydatidina from the small intestine of canids. And also the necropsy or postmortem, we can also see postmortem of the intermediate host or the goat. We can see the development of metacystode in the internal organs. And this is the morphological characteristics of a scolex of Cystosarcus tenucolis. We can see that the scolex is armed with 2 pair of rostellar hooks, and with these hooks, the parasite, they deeply attached to the mucosa of the small intestine, and they imbibe the nutrition from the host. Then treatment, so there is no effective treatment, only surgical removal of the cyst is done, and that is somehow impractical because the affected herbivores or affected sheep and goat, they do not show any clinical signs until and unless the animal is slaughtered. Nobody can diagnose the case, and we can go for giving albendazole therapy to the sheep and goat. And if there is development of any cyst in the vital organ, so that will be killed by the albendazole. And for controlling infection in the definitive host, we can go for combination with fenbendazole and praziquantel that will kill the adult tapeworms in the intestine. Then prevention and control, so reducing stray dog population and restricting them from feeding small ruminant carcasses and offal. Then correct disposal of contaminated carcasses to prevent access of dogs to them. Creating awareness among farmers about proper disposal of contaminated carcass, as it is a disease having public health importance. Then regular deworming of dog with praziquantel. So these are the different prevention and control measures. Then coming to the another metacystode infection, that is the echinococcosis. So echinococcosis is a metacystodal infection which is having the zoonotic significance, and this belongs to the genera Echinococcus species, and the parasite involved is the Echinococcus granulosa that causes a disease condition that is known as cystic echinococcosis. Definitive host are the dog and other canines. The site of predilection of the adult parasite is the small intestine. Here the intermediate hosts are the herbivores and man. So among herbivores, the sheep are being commonly infected, and the metacystode or the hydatid cyst usually develops in the liver and lung tissues. We can see the development of hydatid in the lung and liver tissues. Then mode of transmission, the intermediate host or the sheep and goat, they get infected by contaminated water or forest that has been contaminated with the feces of the infected dogs. The human infection occurs through intimate contact with the infected dogs while playing with the pet animals. Following ingestion, the eggs pass through the wall. They loss in the vital organs, mostly in the liver, lungs, as well as in other vital organs like kidney, spleen, heart, brain, muscles, and bone marrow. Liver and lung are the most common site for development of the hydatid cyst. Then coming to the pathogenesis, so in heavy infection, there will be enteritis or diarrhea in case of dog. But in intermediate host, particularly the disease has got significant importance in case of human, because in case of human being, the hydatid cyst develops in the liver as well as in the lung tissues, and very commonly the liver is the organ where we can see development of hydatid cyst. And this hydatid cyst, it contains the fluid, hydatid fluid, and this fluid is somewhat immunogenic in nature. So there may be sometimes rupture of the cyst, and because of the rupture of the cyst, the fluid from the cyst, as I have told that it is immunogenic in nature. So it will cause allergic reaction to the host, and the host may die due to anaphylactic shock. So sometimes even during surgery, if any of the cyst is ruptured, and if that fluid is fall on the peritoneal cavity, so it will lead to development of the daughter cyst in the organ or in the area where the cyst fluid is fall, and from there a new cyst, new daughter cyst will be formed. And because of fall in the peritoneal cavity, the fluid it induce some immunological reaction or allergic reactions, and that will lead to anaphylactic shock, and death may occur due to anaphylactic shock. Then coming to diagnosis, so routine coprological examination can be done, then purgation with ericoline compounds. So purgation can be done because like other cystode parasite or tinea, so we can see the shedding of the gravid segment. Whenever the gravid segments are full with X parasite, they is to shed the gravid segments in the feces of the host. But in case of Echinococcus, so it is a four-segmented parasite. It is a small parasite, so there is no shedding of gravid segments, but the parasite it usually shed the eggs in the host feces, and or sometimes if we need the adult pressure, we have to induce purgation by introducing ericoline compounds or erica nuts. We can also, we can feed the erica nuts, and the animal will so the defecation, there will be purgation, and we may get the adult parasites, and from the adult parasite we may get the eggs. And necropsy of the visceral organs of intermediate host. So this is considered as a gold standard technique for diagnosis of echinococcosis, mostly in the intermediate host. So this is the liver tissues, and here we can see the development of the hydatid cyst. The cyst, it is surrounded by fibrous tissue capsule, and the entire cyst is being calcified. It is appearance just like cheesy material, and this is the hydatid cyst that develops inside the liver tissues. And we can also, in case of human being, there are some diagnostic technique we can use, like say X-ray, ultrasonography, MRI, CT scan. So these are the different techniques we can use for diagnosis of echinococcosis in human being. And some immunodiagnostic technique can also be applied. Detection of coproantigen by sandwich ELISA, then indirect hemagglutination test, indirect fluorescent antibody test, coproantigen detection by enzyme immunosorbent assay, then immunoblood assay or gel diffusion assay. So these are immunodiagnostic technique can be used, and there are also molecular tools like PCR and DNA probes. So these are being used, but the gold standard technique is the detection of the hydatid cyst in the postmortem of the affected animals, and by detection of the specific Echinococcus ova in the fecal samples. Then control of cystic echinococcosis, we can interrupt the life cycle of the parasite by avoiding access of dogs to eat the raw offal of the small ruminants, then regular deworming of the dogs for tapeworms, then destruction of hydatid found in the slaughtered animals, destroying of stray dog populations. But in a country like India, we cannot destroy the stray dog population, but we can go for mass deworming of the stray dogs. Then general education program for the human beings. Avoid close contact of children with their pets. We should educate the children for maintenance of hygienicity or personal hygiene. We should teach them for the importance for personal hygiene, and we should aware them about the importance of these diseases and how these infections are being transmitted to human. So this type of education we should teach our children, that maintenance of hygiene and sanitary measures. So these are the different control measures for control of cystic echinococcosis. Then finally coming to the treatment and control. So as we have seen that tapeworm infections, they both larval stages as well as adult stages of tapeworms, they cause disease in small ruminants, particularly sheep and goat. In case of larval infection, we can see the dogs, they are the definitive host, and they get infection through feeding of infected offals harboring the metacystose stage of the parasites. So here in this case, we can deworm, we can prevent infection to the definitive host or carnivores through strategic deworming, and thereby we can decrease the egg count and worm load. So praziquantel 5 mg per kg body weight is found to be highly effective against the tinea infections, and dogs should not be allowed to defecate in the grazing land and ranging field. So these are the measures we can use in case of dog for prevention of infection in the dogs. So if we can prevent infection to the dogs, so simultaneously the infection to intermediate host can also be controlled. Then besides this strict entry restriction of the dogs and other wild carnivores inside the slaughterhouse can be done. Then proper slaughterhouse waste disposal should be adopted. Provision of cooked meat and offals. Then avoid feeding of raw offals and meat by dogs. So these measures we can take to prevent tapeworm infection in dogs. So if especially for metacystose infection, because dogs are the definitive cause for metacystose infection in case of small ruminants. So by preventing infection in dog, we can reduce infection load in case of small ruminants. Then periodical plowing and reseeding of pasture helps decrease the infected mite load. As we have seen in case of adult tapeworm infection, particularly in Monesia infection, so we can see the infection generally comes from the grazing land. So periodical plowing of the grazing land is required. Then rotational grazing is also required as well as, so if we can give rest to the grazing land, minimum 2 months or 3 months of time, if we prevent the animals to grazing in that land, even if the infected dog, they avoid the eggs in the feces. So these eggs, they will undergo desiccation. If they cannot catch a intermediate host within a stipulated time, so these eggs will undergo desiccation, they will be died, and there will be no, the land will become sterile, and it is safe for grazing by the small ruminants. So some resting of the grazing land is required, or within that resting period, we can go for cultivating that land or reseeding the pasture land. So thereby, if there is presence of any infection, or in case of Monesia infection, as we see, the oribatid mites, they transmit the infection. So even if the oribatid mite, they become infected with the cystic if they fail to propagate the infection to the intermediate host, so the infection, the cystic will die, and no more infection will be transmitted to the small ruminants. Then secondly, if we can go for strategic and prophylactic deworming of the livestock, sheep and goat, particularly in the late spring or in the early summer, and again in the autumn. So twice deworming, if we can follow, just before onset of monsoon as well as just immediately after monsoon or immediately after flood, if we can deworm, so we can reduce the infection load. So before putting the animals in a grazing land, if we deworm the animals, so because of that, they will less commonly infected, and whatever infection they get in during the summertime or in the monsoon season, so if immediately after monsoon, if we deworm the animal, whatever infection they get, that will be eliminated by second deworming. So by adopting both strategic as well as tactical deworming, we can control the infection in the small ruminant, and thereby we can reduce the infection. Then prevention of reinfestation by regular deworming. So regular deworming we should, but whatever, so to prevent anthelmintic resistance, we can go for routine examination of the feces for presence of ova, and based on the fecal examination report, we can go for deworming of the animals. Then strategic pasture and grazing management, already we have discussed. So control of the grazing land or by rotational grazing, so we can reduce the infection, and for that we can also suggest for zerograzing. So herbivores or especially small ruminants, if we advise for intensive farming, or to say to prevent animals to grazing in the open grazing land, so they will become infected. But if we prevent them grazing in the open land, and if we provide pastures in the farm itself, or if we adopt for intensive farming system, or if we suggest for zero grazing, we can escape from infestation by the different tapeworms, and thereby will maintain the productivity in the animals, and will our economy will be improved. Otherwise, so infection is always available in the environment. We cannot make an environment zero parasite, but we can adopt this type of tactics, especially zero grazing we will advise, because in zero grazing there will be no further infection from the environment, and we can go for production of the animals, or we can increase our activity and thereby uplift our economy, and proper hygiene and waste disposal. So these are the measures we can use it for prevention of tapeworm infection in small ruminants, particularly sheep and goat.